The widespread decline of corals in tropical reefs in recent decades is often accompanied by an increase in algae. These algae compete with the corals and sometimes dominate the reef landscape. Competition between corals and seagrass is expected to increase coral mortality. Some physical, chemical, or microbial mechanisms may directly or indirectly mediate competitions between corals and macroalgae. Such effects can include shading, abrasion, overgrowth, and changes in the coral's microbiome. A manipulative field experiment was conducted to investigate the effect of the proximity of common macroalgae, such as Sargassum polysystem and Galaxaura rugosa, to corals. Attention was paid to the growth, survival rates, photosynthetic efficiency, and microbiome of the coral. It was specifically tested whether algae require an impact on corals in a direct contact or when the corals are in close proximity to algae with a distance of 1.5 centimeters. The study was conducted along the coral coast of Fiji. Branches of the coral Acropora millepora, which were 6 to 8 cm long, were used. These branches were glued into cut necks of inverted plastic bottles. The screw top from the bottle was anchored to the substrate with a nail. Each branch was then screwed into the screw top. After the acclimatization period of 1 month, they were subjected to the experiment for 33 days. For this, the macroalgae Galaxaura and Sargassum were attached to the corals either with direct contact or within a distance of 1.5 centimeters. To keep the algae in position, grids were placed around the coral algae structures. For comparison, control corals were kept separately in a cage without algae. The evaluation of the results after 33 days were based on different procedures, which included pulse amplitude modulated fluorometry, RNA later, and an rRNA gene amplicon analysis for which the DNA was extracted from the coral sample and provided with modified primers. None of the corals died during the 33-day experiment. Compared to the control corals, the growth of the corals in direct contact with Sargassum and Galaxaura was significantly reduced, within a range of 59% and 61%. The corals that were in a distance of 1.5 cm to algae showed growth comparable to that of the control corals. Corals in direct contact with the algae had a significantly lower photosynthetic efficiency than the control corals. The coral microbiomes did not differ significantly in composition, variability, or diversity among the 3 different test groups. The results suggest that the negative impacts from these common macroalgae species are contact-dependent, spatially limited, and the overall impact on the coral microbiome is rare. The relevance of this study is the reduced coral growth of 60% in direct contact with the macroalgae leads to an increased risk of mortality, reduced competitiveness, and fertility of the corals. Determining coral macroalgae interactions are important to understanding and predicting their impact on ecosystem dynamics and reef decline.